Hello everyone, Bismillah Rahman Rahim, Assalamu alaikum. Today's tutorial is a very interesting tutorial for a lot of biologists and biology students and research students. I have thought about it a lot because there are a lot of things that are present in different patches in my YouTube channel, in other YouTube channels, and in a lot of tutorials that how to do RNA-Seq data analysis, whole genome sequencing data analysis, and other analysis. Because this is the day, and this is the year, and this is the time of big data, and there are a lot of big data, and biologists feel like they are way behind from other bioinformaticians or data scientists, and they are not able to handle the data. And if they want to handle it, it needs a lot of programming, it needs a lot of skills, it needs a lot of different resources to be used for. Like for instance, there are a lot of commercial tools that are available, and which can do all the RNA-seq or whole genome data analysis. But for that those are commercially available. They are very expensive, and it's difficult for a basic research student or a basic scientist to use those tools. But there are a lot of ways, and there are a lot of resources that can be used in different from different resources and combine them together and do the analysis. So basically, when you want to do it, you first need to know that what actually is the whole process, because usually the students, they don't know, either they are bad in biology, or either they are bad in computers, and both of these things actually combine together and they complicate their analysis. So when you are good at computers, but you know how to do the programming, and you know how to do the bash scripting, and you process the files as quickly as possible, but you don't know the basically the steps, that what each step means and why this is important. On the other hand, the biologists actually know that they need the reference genome, they want to do the analysis, but they actually have a lot of challenges with the tools, or with the skills, that how they can utilize their skills to get their desired gene of expression data and process the whole files. So this is the whole, I've put this summary here, so that first you need to know that what actually is each step. So obviously, when you are working with the with the sequencing data, RNA sequencing data, uh, definitely whenever you do that, you have some experimental design, and that experimental design is basically that can be to plant genomes, that can be to bacterial genomes, that can be to human genomes or disease genomes. Like for example, there's a most commonly used is cancer genomic analysis, and in the cancer genomic data, what you have, or any rare disorder data, what you have two groups. One is the diseased group, and the other one is the control groups. So so you need one sequencing data from different cancer samples, and then you need the sequencing data of a normal sample, and then those files which you have, they will be in one file, and that file would be a raw file, and that is the FASTQ file. And yeah, so those FASTQ files would be of the cancer data, cancer genomes, and the normal transcriptomes of normal genomes. And these files, these FASTQ files, are generated by different companies and different platforms. Like one of the most commonly is the Illumina, and Illumina sequencing files, and those are the FASTQ files. And these files basically contains all the raw information about the particular data or experimental design you're working on. And these files, the raw files, are very very big files. When you talk about the human genomes, they are more than 10 GB. If you're doing single sequencing, then maybe you have a less size, and that would be 10, around 10 11 GBs. But if you are doing the paired end sequencing, then that would combine the forward and reverse, and that would, the one file of one cancer sample would be around 20 GBs, because depending on the depth and the sequencing depth, or the coverage you are using, like these things you can study separately, that what is a sequencing depth and coverage. And these files, these raw files, are actually, I will explain one by one that these where these files are stored. But first, you go through the whole process first, and then we can do the next tutorial about where these files are located and how you can download it. And so these files are basically stored in different platforms or browsers like NCBI. As in NCBI, you can go and you can find the SRA archive, and in SRA you can download the sequencing files. And same for the, if you go to the Array Express or if you go to the Amble, you can download it from ENA. You can search it ENA and you can see that how you can access the raw sequencing files. So some files are publicly available and some are not publicly available. They have authorized access, but in current world, the amount of data that has been generated, it's, I would say that it's almost impossible that you cannot get a desired experimental data you are looking for in a public platform. You can get easily, but this step, at the raw data acquisition, can be like, no, I think it's a little bit clear to you that you can download it from those platforms. And then the next would be that, obviously, when you have a raw file, then you have to first check the quality of the file. And for the quality of the files, you have to go to you have to use basically a FastQ tool, and that FastQ tool can be used through programming, and that can be used without you having programming skills. I will tell you how. So you can do it by FastQ, like if you want to do one, and there's a multi QC that you can do multiple files together. So it actually tells you that if the quality is good, the read quality is good, or and like what's the GC content, what if is there any debtor contamination or not. So that's how it works. I'll show you in the next tutorial as well, this one. And then you actually, if you have low quality sequences, then you do the trimming. In the trimming, this is just there are multiple tools, and you can use them. Galore is one, Trimometric is another, and it's optional. If there is a poor quality of the sequences, then you go to this. And then when you have the quality raw FastQ file, then you start to do the sequencing analysis. And for the sequence analysis, it's a very simple thing that you have a raw file and you don't know that what is inside it. Then what you do is you compare it with the reference genome. And with the reference genome, all the reference genomes are available on NCBI and Semble UCSC browsers, and you just have to download it. There, and the download after downloading it, you can also see that there are multiple files. They also give you the multiple files, like the annotation files along with the reference genome file, which you use for the sequencing. And obviously, this whole tutorial and lecture series would be only for those biologists who cannot use programming. So I'm not going into that, how you compare the reference genome and how you download it and how actually you can make a input file for the reference genome and then you compare it with the raw files you have. So we will skip that for now. And then you do a lot of other things, like if when the FastQ file is then converted into the BAM file, and the BAM file is actually the binary alignment. From SAM, you convert it into BAM, and that is done after the when you align it with the reference genome. And then basically, you now the file is ready, and and the file is ready, like for the align. Sorry, if I explain a bit more, is that you do alignment by different tools, and that is star, top hat. There are a lot of tools, and then after doing the alignment, you use SAM tools, Picard tools to convert those files into BAM files. And then after, when you have the BAM file, which is aligned file, indexed file, and that is compared with the reference genome, what then we look for the counts. Basically, that count means that how much the each gene is amplified or highly expressed or low expressed, in simple words. But what actually you get by different tools like HT-Sec count, feature count, Salmon, these tools, what they give you is the count reads per gene or the transcript. So total number of count of a gene. So it means that this is the actually the first primary information about the the gene expression data, that how much each gene count of each gene is reported, right? And that can be done by multiple tools like feature counts and so on and so forth. These tools, if you want to do through programming, I have made the tutorial like how you can use these different steps and how you use different commands to do this feature count analysis. Also for how to convert SAM to BAM and how to convert FASTQ to SAM BAM. And after doing that, you do normalization. And after normalization, there are different methods like doing the normalization of the these counts and doing the differential expression. That is like different expression analysis. There are multiple packages for that. DSEQ is one package. DSEQ2 is a modified package of the DSEQ. Lima, LIMMA, Lima is another package that is used to do differential expression analysis. And this differential expression basically means that the difference of expression between the two groups, and that two groups are those two experimental design you started from the beginning, like tumor versus normal, disease versus non-disease, one plant versus another. So that is the design you made in the beginning. And after doing that, multiple that you get the differential expression analysis, and then doing the differential expression, you do that, which one are the most significant ones, which are not the significant ones, and for that you check the p-values, do multiple testing corrections, and then you get the most significant genes from starting from the raw FASTQ file to the after, and then pre-processing, normalization, and then the differential expression, and then you get the most significant genes that are the genes which might be of your use or interest, that you can do after looking into the genes. And then when you have that process file, and that process file actually is just of couple of MBs size, and now you can see that you started from hundreds of GBs of files, and now you ended up with a very small 1, 2, 3, 4, or 10 MBs file, and that file is enough for which contains all the information about the differential expression. And then when you have that file, then you can do multiple analysis like functional enrichment analysis, pathway analysis, like the KEGG pathway and uh, reactome pathway, and all types of pathway you are looking for. That we will discuss later. And there are a lot of tools, freely available tools, which can be used to do this whole analysis. So first you could go through this, and then when you know about the whole process, then we will start from the beginning, that like how you can download the data, how you can look for different files, and how these files can be used for this whole analysis. Again, keep that in mind that we will not use any programming, and we will particularly focus on the steps. So if you and another thing, that if you want to do the whole process yourself, I have a lot of tutorials already. Here I will only explain that experiment, like for example raw data acquisition, from where you can download it. I will show you the example that here is the file, this you can download. Then if you have a downloaded file, how you can do the quality control, for fast QC, fast QC, yeah. And then how you can do reference genome only through non-programming approaches, or you can say the biologist approaches. So thank you very much. I think that's enough for now. And the next tutorial will be about the acquisition of the raw files. Thank you. Bye bye.